Typically, when we talk about cancer genetics, we discriminate two types of genes or two types of mutations. Oncogenes, which are genes that, when mutated, cause cancer, and tumor suppressor genes, which their function is to suppress cancer, and when they misperform, then it's kind of like the brake on cancer is set apart. However, there's another set of genes that play a role in cancer genetics, and that is protective genes. So there's certain allelic variants, there's certain mutations, there's certain genomic changes that correspond to a lower chance of getting cancer irrespective. And basically, the protective genes would be genes that would confer an individual with lower chance of getting cancer as compared to the general population. There aren't a lot of those that were found, but the ones that have seem to suggest a paradigm. So here's a few. So there's a whole bunch of genetic association studies where people took very aged individuals, and looked for genetic associations that can predict longevity. And there's multiple of these studies, and they predict different genes, but one common denominator that continues to emerge is the link to this pathway called the MTOR pathway, the insulin down to the MTOR pathway. So mutations that lower the activity in this pathway seem to increase lifespan and decrease cancer risk. Now, the link of this pathway, the mTOR pathway, to lifespan and reduced cancer risk is not just genetic. It also comes from pharmacological manipulations, which makes it very interesting. So, for example, here, this is a study where they took mice models that have mutation 53. Here, it's not mutation, it's complete knockout, heterozygous knockout. And these mice get cancer at very high rates. When these mice were treated with a drug that blocks this pathway, cancer frequency was reduced. And this paper is one of many, many, many. Here's another one. That's a review. And here's one specifically in Leaf-Fermeni. So what these people did is they took mice models of Leaf-Fermeni. So these are mice that have 53, but these 53 have point mutations that resemble the Leaf-Fermeni phenotype. And they inhibited this pathway indirectly, and again, they saw a reduction. They claim completely no cancer, but they saw a reduction in cancer rates. So this is kind of seems to be a pervasive story, that inhibiting this pathway seems to prevent cancer onset. And this is not even exclusive to the context of 53. We even have papers showing this with transgenic HER2 mice that have inactive oncogenes, et cetera, et cetera. So then the question is, instead of targeting 53 or killing cancer cells, can we somehow prevent cancer? And this is particularly important for Leaf-Fermeni patients. So now let's try to think, and let me tell you one more thing. Like I said, I'm kind of a newcomer here to this field, so my talk will maybe present more questions than answers, but I will try to present some answers. So let's look and think at what's going on. So we have this drug, Rapamycin, and it inhibits this pathway, mTOR, and that prevents cancer. Now, how can it do that? Well, one thing we do know is rapamycin, this drug, is very ineffective in treating existing tumors, but it is very effective in preventing cancer. And taking these two considerations alone, it seems to be that what rapamycin, or what this pathway is targeting, is not the cancer cells itself, because otherwise it will also be effective against the tumor itself, but rather it targets the environment that allows the enabling conditions, the environment that allow cancer to form in the first place, which kind of turns our attention to the stroma. Okay, so here's the first experiment we kind of did to test this, and this is in collaboration with David. We took Fireblast from Leaf-Fermeni patients, and we took mice, and we injected the mice with cancer cells, with normal fireblasts or fireblasts with coming from Leaf-Fermeni patients. And here's the result of this. So you see 3 lines. This is tumor volume as a function of time. The 2 bottom lines are cancer cells that are injected with no fireblasts, or cancer cells that are injected with healthy fibroblasts, while the red line shows you cancer cells that are injected with fibroblasts from Leaf-Fermeni patients, and you see the tumor grows much, much more aggressively, which suggests that somehow these fibroblasts, even though they're not transformed, they're not cancerous, they support the cancer in some way. And this kind of homes our attention into a different thing. Instead of looking at the cancer, the tumor, we're looking at the environment that allows it to become. Okay, so now that we kind of zeroed in on a target, how are we going to explore it? So what we're looking for is some kind of signaling in the pre-malignant niche that we can change to disfavor cancer onset. And so we want to kind of, we don't know the answer, so we want to try to look at as many things as possible. And this is, again, Varda told you towards the end that today what we're doing is trying to collect, or many people are doing, trying to collect a lot of information from patients. So this is one approach. So this is, again, a collaboration with David. And this collaboration builds on a very unique resource. So we have an assembly, a library of patient-derived fibroblasts. Each fibroblast is collected from a different patient. Each patient associates with a different genetic background and a different point mutation in p53. And now we can look at all various properties of these cells and try to identify properties that are predictive of a microenvironment that prevents cancer. Now, how are we going to look at what would be considered fibroblasts that prevent cancer? Well, we can think of three sources. One is a healthy fibroblast. Second is a fibroblast treated with rapamycin, because we know that rapamycin prevents that. And third, we can even look at it for many patients who happen to have very low cancer rates. But having all these comparisons, what are we going to look at? And so we started with three different types of phenotypes. One, we're looking at these fibroblasts and looking at intracellular signaling. And we're trying to say, are there signals in these fibroblasts that don't appear in the non-protective fibroblast? The second is morphology. So, and this I'm going to talk a little about, but here what we're asking is, can we see a difference in how they look, the protective fibroblasts, compared to others? And the third is paracrine influence. Now, what does paracrine influence mean? These fibroblasts somehow influence the cancer. So they signal to the cancer cells to either disfavor their survival or disfavor their growth. So can we quantify that too and find differences? And so now I'll give you a little bit of how we do this. This shouldn't have been here. So most of our tools is quantitative microscopy. We do a lot of microscopy really quickly, and then we do image processing. We use a computer to identify cell borders and get multiple readouts per cell. And here what you see is what is one well one population of cells. Each dot is one cell measured for three different properties. And from that, we calculate the distribution. And this gives us a picture of the signaling state of a cell. Okay, here's, it kind of shows, it's a very informative picture, and we can do a lot with it. I'm going to skip forward. How can we estimate what patient-derived or what sick fibroblasts look like? And so, since we a priori do not know, so we do the following game. We invent a lot of features of cellular appearance. For example, to what extent is the cell circular or elliptoid? That's called eccentricity. Another one is to what extent is the cell regular? And so we invent as many of these morphological features as we can imagine, and then we have a series of features per cell, and then the computer looks for discriminatory features. I'm going to stop here, and then the last thing, and this is a collaboration with the Weizmann Institute. Yeah, Varda knows. So I want to introduce you to Vid Straussman, which is a good friend of mine, and a very inspiring scientist as well. And here we borrowed from Ravid because really what we want to measure, so we quantify how these cells look, we quantify the signaling in these cells. Now we want to quantify how they influence signaling in the cancer cells. And to do that, we borrowed a system that Ravid established. It's called a signalome. I'm not going to go deep into it, but the basic idea is we are co-culturing the cancer cells with the patient-derived fibroblasts, but we can discriminate, so they're all sitting in the same dish, the cancer cells and the fibroblasts. But we can discriminate them because they have different colors, because they're fluorescently tagged in different ways. And then we can look at signaling in the co-cultured cancer cells. Now, there's more to this, but I'm going to leave it at this point. So we've basically, up to now, collecting a lot of information on patient-derived fibroblasts, that when we're trying to find how they influence the microenvironment, the pre-malignant microenvironment, to disfavor or favor tumor onset. So that's our goal. So I'm going to take a step back for a second. So you understood kind of what we're doing. So now let me kind of give you a bird's eye perspective for a second. And so how does rapamycin prevent cancer? And we said, okay, rapamycin must prevent cancer by somehow targeting signaling not in the cancer itself, but in the surrounding environment, in the microenvironment, right? But how does it do that? And that's a very big question. But when you think of what rapamycin or MTOR do, they don't just prevent cancer, like I told you, they also extend lifespan even in healthy patients. And on a molecular level, if you look at the real function of MTOR, what it does is it decreases cell size and decreases protein, or mTOR inhibition decreases cell size. mTOR itself basically generates protein mass. Okay, it makes the cells grow. So now the question is, why is this pathway linked to these three phenotypes? But the question becomes even more bizarre when you think of it, because it's not just that mTOR extends lifespan and shrinks cell size. It has been doing that for basically all of eukaryote evolution, from yeast to worms to mice to humans. So this basically says that while we're looking at this question in a very narrow context of cancer, the question itself is fundamental, deep, and it might hold some very fundamental constraint on lifespan itself. And so I want to, before I get to the following slide, so we came to this subject from studies on cell size, and so we were very familiar with mTOR because it influenced cell size, and this is kind of where our mind drifted. But then we started collaborating, and this is another very inspiring scientist and friend, Yuval Dor from the Hebrew University. And I'm going to show you one slide that Yuval generously included me on this research. So Yuval studies the pancreas and pancreatic beta cells and insulin and diabetes. But by way of a series of coincidences, which I'm not going to go through with you right now, he started collaborating with the Jerusalem Zoo and looking at size of cells of different mammalian species. And he came up with this plot. And he started collaborating with us to try to figure out what this plot exactly means. So let me just walk you through what you're seeing here. And I won't even try to understand why you're seeing what you're seeing. But here's the data. So basically, every dot in that plot is the size of cells in one animal species. The x-axis. And the y-axis is the lifespan of that animal species when living in captivity. So these animals don't die from predators, etc. And so you're asking, okay, first of all, this suggests that there's something very fundamental here. But now you'll ask, what's the connection to Live for Many, right? And so the main pathway that drives growth in cell size is the mTOR pathway. So we thought, hey, maybe regulators of mTOR, regulators of growth, also associate with longevity and cancer prevention. Now, before this whole thing started, we performed all these different drug screens and other screens to identify novel regulators of cell size. And we discriminate two types of size regulators. One is things, genes or drugs that change the size of cells, but do not influence the feedbacks that maintain it at its place. And then we have a whole set of other perturbations that really throw cells off their regulation. Let me just for intuition walk you through this plot. So, because you've seen it before, I should have said it before. So the blue dots are controls. And these controls, they have a certain, so these are cells, they have a certain rate at which they increase their mass and a certain cell cycle length. And when you add a drug or genetic perturbation that, for example, increases the amount of time that they grow, they will compensate by decreasing the rate which they grow to maintain size constant. And so these type of drugs or genes basically break this coordination. So somehow they affect the homeostatic control of size. And when we compared the hits from our screen with known lifespan regulators from the literature we saw dramatic overlap. Here's the actual overlap. So what you see here, these are hits from our screens. And you see that the percent of known influences on lifespan decreases as you move to perturbations that increase and increase cell size. And again, this is not all size regulators. This is a subset where we remove the ones that perturb the feedbacks. All right. So coming back, so where do we stand? And now I have just a few minutes to finish, but it's actually, I'm in good time. So so we started off by saying that mTOR prevents cancer. It also extends lifespan in some way. But the lifespan is a very complicated question. The cancer is somehow simpler. And so by preventing cancer, it's pretty clear, because it's not killing the cancer cells, that it somehow changes the microenvironment where the cancer originates in the first place. And so I gave you this overview of a research direction where we're taking patient-derived fibroblasts and we're measuring how mTOR influences them, or how mTOR inhibition influences them across various traits, and try to identify a signature that might be predictive for drugs that prevent cancer. And the signature that we're building, I told you, has 3 arms. One, we're looking at the signaling of the stromal cells of the microenvironment. Second, we're looking at morphology, at how they look. And the third thing is we're looking at paracrine influence by collaboration with Ravid Straussman from the Weizmann. We're looking at how they influence co-cultured cancer cells. So that's kind of the direction, and I'll show you initial results from this direction. Because some of them even surprised us. So starting with the signaling, so like I showed you before, we're using microscopy, high content microscopy, to capture multiple signaling events per cells, and we're getting these very large distributions. What I showed you before was data collected that characterized cell cycle progression and cell size. But right now, we're taking that same approach and bringing it a bit closer to relevance to leaf for many. So here we are measuring mTOR activity, and we're measuring a cell's response to DNA damage. Again, it's immunofluorescence, so you see the cells are labeled for, here there's 2 colors. Green is the amount of DNA damage as assayed by phospho-gamma H2AX. And the red is mTOR activity assayed by some downstream effector of mTOR. And you can see, and so what we're doing here is we're adding, I don't know if this is a drug or radiation, I think it's radiation here, but basically we're irradiating the cells which causes DNA damage, and then you see them becoming green because they're expressing or they're activating the DNA damage response pathway. But you also see that the redness decreases, and that's because the DNA damage activates p53 and p53 inhibits mTOR. And again, here's a more complex look at the same data. So what you're seeing here, this is, and now, again, this is a glimpse to the complexity of the data that we're dealing with. So let me start here. This is a human-derived Fireblast, but this is a healthy individual, not a leaf from any patient. And what you're seeing here is the distribution. So the black, it's a distribution of mTOR activity and DNA damage response in a population of cells. And there you see red, blue, and green. Red, black, red, and blue. I'm slowly losing my mind, but it's okay. So black, red, and blue. Black is the unperturbed population. Red are cells that are treated with rapamycin. It's a drug that inhibits mTOR. And blue are cells that are treated with doxorubicin, which is a drug that causes DNA damage. And you see that as you would predict. So when the blue ones, you see that they have more DNA damage and less mTOR activity. And then the red ones, rapamycin inhibits mTOR, so you see less mTOR activity. So this is kind of as you would expect. But now, here's the neat thing. This and this is the same plot, but it's generated not from healthy individuals, but from patient-derived fibroblasts from leaf from many patients. And these 2 patients associate with different mutations. And you can clearly see differences. For example, this patient has much stronger mTOR activity, or high levels of mTOR activity as compared to the wild type, while this one has a lower mTOR activity as compared to the wild type. Okay, so we can start now profiling these patients based on signal transduction cascade in this kind of multivariate approach. So that's that, and here they also differ in the DNA damage. I'm going to skip this. Now, we also said that we can see how cells look using these morphological features. And here I have to tell you, I myself was surprised. So basically, to remind you, what we did is we built all these metrics that quantify different aspects of cell shape. And then we tell a computer whether, based on the metrics that we give it, can it discriminate fibroblasts collected from, example, healthy individuals to leave for many patients. And so, and I didn't think it would actually be this good. So what you see here, this is the computer. So we tested a whole bunch of different discrimination algorithms, and this one worked the best. It's called random forest, and it has a 98 or a 90, depending on over 90% discriminatory power between human fibroblasts collected from healthy individuals to human fibroblasts collected from leave for many patients. But our discriminatory power goes even deeper. What you see here is the ability to discriminate fibroblasts from different leave for many patients based on point mutation. So you collect patient-derived fibroblasts from two different patients that differ in the governing mutation in the p53, and we can discriminate just based on how these fibroblasts look, right? And the assay can even improve with time because we can image more morphological features. Okay, and so this is kind of, I'm running out of time, so I'm just going to kind of lay out where we're going with this. And so we're really moving in two directions. One is we're studying how MTOR lifespan, and second is cancer prevention. But in the context of cancer prevention, in the more narrow window, we are basically going to use these assays of paracrine influence, signaling, and morphology to identify signatures that correlate with a protective microenvironment, a cancer-preventing microenvironment, and then we can perform drug screens to look for rapalogs, if you want, drugs that mimic this function. I guess, and with that, I'll end. I have to thank the team. So this is the group, but I want to mention a few people in particular. I have to actually start with this. So these are the three people who did most of the work. Camila presented a poster a few days ago, and she's carrying the Leave for Many project forward. Serial Tan is collaborating with the Toronto Zoo to collect more and more measurements and trying to look at the aspects of this into lifespan. And Teresa Gebert is a statistician from Carnegie Milan that's helping us with the machine learning to try to extract the signatures that are predictive. And this is the rest of the group. And these are also Jeremy Jenkins, Yuan Wang, and Mark Hild are collaborators from Novartis Institutes for Biomedical Research from Boston, and they will be helping us to perform the screens once we establish the assays. And that's it, and thank you.